Hello, I'm Dan Limbach from PharmaVoice, and I'd like to welcome you to today's event entitled Galectin-3, Enabling a New Pharmaceutical Paradigm in Treatment of Heart Failure, brought to you by PharmaVoice and sponsored by our good friends at BG Medicine. We have a very nice crowd today. Thank you for investing the time to attend. We'll certainly do our very best to make it worth your while. Before we get started, I would like to take a moment to acquaint you with a few features of our web seminar technology. If you are experiencing problems with the technology, please press the F5 button on your keyboard for PC users or Command R for Mac users. Or close your window and relaunch the presentation using the other media player option you saw when you logged in. You can also visit our webcast help guide by clicking on the Help button near the lower left corner of the slide window. Our help desk is also available throughout this event by calling the number on the help page. The web seminar is designed to be interactive between you, the audience, and the presenters. You can participate in the Q&A session by asking questions at any time during the presentation. Just type your question in the Ask a Question text area in the bottom corner of your screen, then click the Submit button. You may enlarge the slide window at any time by clicking on the Enlarge Slide button located near the lower left corner of the slide window. The slides will advance automatically throughout this event. You will receive a link to the archive of this webinar in about a week, so you can view it at your leisure as an on-demand presentation. We'll also be sending you an email asking you to complete a brief post-event survey. Please take a moment to complete the survey as it will help PharmaVoice and BG Medicine plan future web events. Now that we're through with the housekeeping details, let's dive into the content. It's going to be an interactive session. You'll participate in live polling questions and use a real-time tool for submitting questions for our live Q&A session. You're going to be seeing and hearing a great deal of useful information today, and you don't have to memorize everything during this live event. We will send you a link to the on-demand archive, as I mentioned before, in about a week, so you can review the event at your leisure. There's a large body of research associated with Galectin-3. A new report comes out about every 36 hours. We encourage you to stay connected on the topic of Galectin-3 via social media. The Twitter ID is @Galectin3. You can also connect to a brand new Facebook interest page using the word Galectin-3, and you can see those on the screen. We have a great panel to help us understand the issues and navigate through the challenges presented in today's environment. Let's learn who our thought leaders are. It's quite a distinguished panel. First, we have Bertram Pitt, MD, FACC, Professor of Medicine Emeritus, University of Michigan School of Medicine, Division of Cardiology. We also have Rudolf de Boer, MD, PhD, Associate Professor in Cardiology at the University Medical Center in Groningen, Department of Cardiology. And finally, Peter Muntendam, MD, President and CEO of BG Medicine. Peter's going to start things off today. Peter, you have the floor.
Dan, thank you so much, and I want to thank the audience for participating today. What we wanted to do today is introduce you to Galectin-3. We have been developing Galectin-3 as a diagnostic, and in the process realized how important Galectin-3 can be to enable a new paradigm in the treatment of heart failure, and we'll tell you more about that, and we want to share that with representatives of the pharmaceutical and biotech industry. Very quickly about BG Medicine, we set out to create a new type of diagnostic company, a company that discovers and develops new content, high-value content, meaning high-value new diagnostics, and then bring it to the market in partnerships with leading diagnostic companies such as Abbott, Siemens, Alere, and bioMérieux, for example, for our Galectin-3 test. Each of these companies is currently developing a Galectin-3 assay for their automated instruments, making Galectin-3 accessible for widespread clinical and research use. In our discovery and development, we focus on major unmet medical needs. We believe that new diagnostic holds the key to better medicine for less. We have a pipeline of what we believe to be transformative diagnostic products in the area of cardiovascular disease, and our first product, Galectin-3, was cleared by the FDA in 2010 as the first new diagnostic, cardiovascular diagnostic cleared in 5 years. We continue to discover new diagnostics. For example, we are partnering with the Framingham Heart Study, the first such collaboration by the Framingham Heart Study in the 60 years of their existence, to discover new blood tests for use in patients at risk for cardiovascular disease. For further information about the company, I refer to our website, www.bgmedicine.com. I'll talk about the objectives of the meeting today. I want to introduce you to galectins. I want to talk briefly about how important heart failure is as a medical condition. I want to talk about the particular subform of heart failure. We think that heart failure is transitioning from a condition with an emphasis on the function to an emphasis on underlying disease, very similar to what happened in other conditions such as liver disease, where the focus shifted from a failing function focus to an underlying disease cause focus. And we think that galectin-3 is a catalyst to a similar transition in the area of heart failure. The area where galectin-3 plays a role is cardiac fibrogenesis, progressive fibrosis of the cardiac muscle, and the opportunity to disrupt that galectin-3 pathway, galectin-3-mediated pathway. I'll talk about the future of drug development in heart failure and the role of galectin-3, and then the opportunities for collaboration. Very briefly about galectins. So there are two key characteristics that are part of the definition of galectins. They bind specifically to certain carbohydrates. This is unrelated to disorders of carbohydrate metabolism, for example. It's nothing to do with diabetes and other carbohydrate metabolism disorders. No, those are very specific carbohydrates that bind to the protein, and they also have a particular sequence of their carbohydrate recognition domain in common between the galectins, and approximately 15 galectins have been recognized as far, characterized as far. The very interesting aspect of these galectins is that they have a carbohydrate binding site whereby the carbohydrate can actually turn the protein on or off. So they have a built-in carbohydrate switch, something that was poorly recognized until the galectin research emerged, namely that proteins have important biological functions, but in those functions are modulated by carbohydrates. Galectin-3 in particular has drawn most of the attention in the scientific investigations. There are approximately 1,500 articles about galectin-3 to date. There's about one new article approximately every 36 hours that references galectin-3, and a lot of truly innovative research is being done in the area. When you review the literature, you'll see that there's very rich literature on the intracellular role of galectin-3 and an increasing body of literature on the extracellular role of galectin-3. And galectin-3 is actively secreted by certain cells, including the macrophages, and it reaches the extracellular space, is responsible for certain biological functions in the extracellular space, and through that mechanism also reaches the bloodstream where increased levels can be measured. It's now an honor for me to introduce Dr. Bertram Pitt, Professor of Medicine Emeritus at the University of Michigan. Professor Pitt, or Dr. Pitt, has been involved with the role of fibrosis in heart failure and the treatment thereof for over 2 decades, and in a tireless effort has drawn attention to this very important medical problem, including his role in the large clinical trials that were conducted in the space, in particular the emphasis study, which was probably the most important study in heart failure that reported last year. With that, Dr. Pitt, please go ahead.
Well, thank you. It's a pleasure to be here. And I think you're all familiar with the data on the slide talking about the statistics of heart failure, what the economic burden is, and obviously this is one of the most rapidly increasing areas of cardiology because of the aging of the population, the epidemic of obesity and diabetes, and has tremendous economic consequences that I think you're all very familiar with. On the next slide is a schema of the renin-angiotensin-aldosterone system, which I think you are also very familiar with. But I'd just like to point out that stimulation of the AT1 receptor results in the adrenal production of aldosterone, And many people have thought that when they give an ACE inhibitor and block the conversion of angiotensin 1 to 2, or give an angiotensin receptor blocker and block the AT1 receptor, that they would block aldosterone production. But that turns out not to be so. No matter how much of an ACE inhibitor or an ARB with a combination, there are other stimuli and an escape of aldosterone. And we originally thought aldosterone was important in heart failure for sodium retention, but now we know it's important for a whole variety of other reasons, and including fibrosis. And aldosterone, as you can see on this slide, is linked to the production of galectin-3 through osteotomy. And once galectin-3 is stimulated, there's an increase in fibrosis, and aldosterone blockers block the mineralocorticoid receptor and block the production of aldosterone through the galectin-3 signaling pathway. Now, why fibrosis is important is there's increasing data suggests that myocardial fibrosis is the critical factor determining the transition to heart failure, the progression of heart failure, as well as sudden cardiac death. Once you have myocardial fibrosis, there's electrical immogeneity and gap junction dysfunction, which predisposes to sudden death. So both the progression of heart failure and sudden death are clearly related to fibrosis, and this is clearly emerging as one of the most important pathways to target in the future. Now, if we move on to the next slide, this is just a schema of patients with heart failure that points out that not everyone with heart failure has evidence of ongoing collagen formation. I've been involved in the RALS trial of spironolactone and the EPISIS trial of eplerenone, and most recently the EMPhaSIS heart failure trial of eplerenone. And in all of these large-scale trials in patients either with severe chronic heart failure due to reduced ejection fraction, heart failure with reduced ejection fraction post-infarction, or most recently the EMPhaSIS heart failure trial in mild symptoms with a heart failure with reduced ejection fraction, aldosterone blockade has reduced total mortality, and in fact, in the recent EMPhaSIS trial, also total hospitalization. But what was striking from the RALS trial is that just about all the effect of the aldosterone blocking agent on total mortality was in the subset of about 50% of patients who had ongoing collagen formation. In those people without evidence of ongoing collagen formation, there was no significant benefit on mortality. So clearly, if one could identify those patients with ongoing collagen formation, one could sort of, let's say, personalize medicine and target the therapy to those people who really need it and avoid it in those people who don't. And as you know, although aldosterone blockade with spironolactone and eplerenone has very effective, there is some incidence of hyperkalemia, and if we could target this more effectively and avoid giving it to people who don't need it, and just give it to the people who need it, I think this would be a major advance. We'll go on to the next slide here. And this is, before I get there, I just want to emphasize again the importance of heart failure and fibrosis. But now we have this polling question, and Dan, do you want to take this over?
Absolutely, thank you. Yes, we want to ask our audience the first of 3 polling questions, and this one is, do you have any knowledge of galectin-3 prior to this webcast? And all you have to do is click the little circle next to the yes or the no, and click the black submit answer button. So while we're waiting for you to chime in, Dr. Pitt, what do you think the role of fibrosis will be in the future from a healthcare perspective?
Well, I mentioned already the major trials in heart failure and reduced ejection fraction, but I'm the principal investigator of the NIH TopCat trial. We're investigating the role of aldosterone blockade in patients with heart failure and a preserved ejection fraction of diastolic heart failure. And clearly there, fibrosis is the critical factor in the transition from hypertensive heart disease to heart failure. And there's good evidence that early in hypertension, diabetes, obesity, fibrosis actually appears before left ventricular hypertrophy or the organ is damaged. Post-infarction is obviously damage and you have reactive fibrosis, but in situations like hypertension, diabetes, and obesity, the fibrosis is one of the earliest morphological manifestations and is responsible for progression of heart failure and sudden death. And we're just about near the end of recruitment for this NIH TopCat trial, which should have results in 2013. But more importantly, I think there's emerging evidence that the real future for aldosterone blockade and other therapies is not the treatment of heart failure, but the prevention of heart failure. We believe that it will be possible to take high-risk patients with hypertension, diabetes, or obesity, and by giving them antifibrotic strategies such as an aldosterone blocker, we can prevent the transition. We'd rather not wait until there's irreversible damage, and we think that by intervening early in patients with a metabolic syndrome or diabetes or hypertensive heart disease, that we can stop this progression. And clearly that would have tremendous economic implications and opens up a tremendous patient population for the future. So I think that's where the future is, and I think the future is very bright.
Thank you, Dr. Pitt. Let's see what our audience said in response to our first polling question. All right, I'm going to push these results out. And 69% of our audience actually didn't have knowledge of galectin-3 prior to the webcast, and that left 30% that did. So it's good to know that there are a lot of people are interested in this and they're looking to learn from it, and it's also good to know that we've got some people with some knowledge of it already that are hopefully looking to gain more information. Okay, let's move on. And
I'm going to Dr. DeGoa now.
Yes, thank you very much for the invitation, and a very good morning or afternoon for you from the Netherlands. It's a pleasure for me to continue and share some preliminary and very exciting data on the potential role of galectin-3 in fibrogenesis, and as it says here in the slide, this might provide an opportunity for attenuating the development and progression of LV dysfunction and heart failure. So the current slide shows you how galectin-3 was associated with heart failure. The first observation that galectin-3 is involved in heart failure was made in a rat model, the so-called RAN2 rat, which is a transgenic rat that overexpresses murin renin and as a result has hypertension with end organ damage, including heart failure. And in the right upper panel, you see the natural course of LV dysfunction and heart failure of these rats. You see compensated as indicated by the arrow, and then later on, over the course of several weeks, some rats remained compensated, indicated as comp, whereas other rats go into overt heart failure. So when we took out the halves of the compensated and the compensated rats and the rats with heart failure and compared them for gene expression profiles, it was observed that galectin-3 emerged as the most overexpressed gene in failing functionally compensated hearts. So from this, the hypothesis was generated that galectin-3 might be a mediator of cardiac fibrogenesis and might result in heart failure development and progression. So in the office of this article in 2004 went on and used recombinant galectin and infused it into the pericardial sac of the rats. And they noticed that this is associated with marked collagen increase, as can be seen in the left panel, and it's also associated with reduced cardiac output. So in other words, the accumulated collagen results in a deficit in terms of LV function. And they went on, as you can see here in the next slide, to corroborate their findings, translating the animal data into human tissue observations. Here as tissue specimens, they were taken during aortic valve replacement, and that generally there was an increase in galectin-3 expression, but this was more outspoken in patients with decreased ejection fraction, so in other words, in patients who had damaged hearts. So in the next slide are some preliminary data generated in our own laboratories. Because the RAN2 rat is, of course, an awkward model of heart failure. It's a transgenic rat. We compared the mRNA expression profiles of rat hearts from RAN2 rats with post-MI heart failure, which is really the most common etiology of heart failure. And we observed that only a small subset of genes was consistently upregulated, including hallmark genes that are very strongly associated with heart failure, for example, atrial natriuretic peptides. But this subset of genes also included galectin-3. So we observed consistent upregulation of galectin-3 in various models of different etiology. So I think this data underscore that galectin-3 is very generic to the onset and development of heart failure. So in the next slide, we try to obtain further insight in the pathways of galectin-3 biology, and we performed pathway analysis. It turns out that galectin-3 is very central in the fibrotic process. It is regulated by osteopontin and released by activated macrophages into the extracellular matrix. Then it turns on quiescent fibroblasts into myofibroblasts, and in turn this results in fibrogenesis. We observed it has not intrinsic inflammatory properties. Inhibition of galectin-3 does not affect inflammation, but it does reduce secondary fibrosis. In the next slide, you see an experiment performed in mice, wild type on the left upper panel, and in mice that lack the gene that encodes galectin-3, galectin-3 or galectin-3 KO mice. And galectin-3 has been investigated in post-inflammation fibrosis models, specifically hepatic fibrosis and renal fibrosis, which was elicited by unilateral ureteric obstruction, or UUO. And it is from that study that you see the results displayed in the right panels. And it can be appreciated that in the wild-type mouse upon UUO, there is marked collagen apposition, as can be seen in panel C, with increases in collagen mRNA expression, but this is attenuated in the GAL3 KO mice, so mice that lack galectin-3 have less outspoken fibrotic response. That's almost absent, in fact. So these studies and other previous observations made us do the following investigation. So we questioned whether disruption of the gene that encodes galectin-3, again using the galectin-3 KO mice, prevent the development of myocardial fibrosis and subsequent LV remodeling. We employed two well-established mouse models of LV remodeling, pressure overload by constricting the thoracic aorta, it's called TAC, or angiotensin-2 infusion by osmotic mini-pumps. And furthermore, we used again the RAN2 rats, and we asked if pharmacological inhibition of galectin-3 would ameliorate LV remodeling in this rat model. So again, here's the animal model cited, galectin-3 KO mice with littermates, and transgenic rats, the RAN2 rats, with SD as genetic controls. And we use the models of left ventricular remodeling by transverse aortic constriction or angiotensin-2. So endpoints were measured using Miller catheters, indwelling catheters with which the invasive hemodynamics can be measured, and echocardiography. We performed PCR and Western blotting to measure collagens, galectin-3 expression, and other expression of other genes. Performed immunohistochemistry to score fibrosis. So here you see the results for mouse studies. In the left upper panel, you see the echocardiography results. CALM stands for control, angiotensin-2 stands for angiotensin-2 infused mice, and TAG for mice that underwent the transaortic constriction. And as you can see, the white bars are wild types of normal mice, and the black bars are the mice that lack the gene that encodes galectin-3, GAL3, KO mice. So control mice generally have normal fractional shortening, which is a measure of systolic function, of contractile function. But both of our perturbations, both angiotensin-2 and TAC, are associated with decreased systolic function, and this is attenuated in the GAL3 KO mice. In other words, our perturbations cause less damage in the GAL3 KO mice that seem to be protected from myocardial damage due to our perturbations. In the right upper panel, left lower, and right lower panel, these are measures of invasive hemodynamic, left ventricular and diastolic pressure, the tau, which is the relaxation constant, and BPDT, which is a measure of contractile LV relaxation. And as you can see, all of these factors are significantly increased or decreased by perturbations, but to a lesser extent so in the GAL3 KO mice, which suggests that the lack of galectin-3 is associated with attenuation of alveolar remodeling. So in the next slide, you see we try to find out what mechanism might underpin this observation, and we look for fibrosis. And in the histological picture on the right side of the slide, you appreciate that in the wild-type mouse under control circumstances there's no fibrosis whatsoever in the heart, but with either angiotensin II or with TAC there's marked increase in fibrosis, which is almost absent in the GAL3 KO mice, and that is quantified in the bar graphs on the in the B panel. So as you can see, again, there is a clear reduction in fibrosis in the mice that lack the GAL3 KO gene. So now we turn from mice to rats. This is the result. These are the results from the rat experiment. In the left upper panel, you see SD control, so that's SD for Sprague-Dawley, that's a normal rat. And then there's 3 groups of RAM2 rats, either treated with control with vehicle, or with our GAL3 inhibitor, or with ACE inhibitor, which is established therapy in heart failure, and it has known antifibrotic effects. So again, in the left upper panel, you see that there's clear systolic dysfunction, contractile dysfunction in the RAM2 rat that can be abolished by treatment with the GAL3 inhibitor or the ACE inhibitor. On the right upper panel, you see what's happening to the contractile function over time. At baseline, more or less comparable contractile function, but over time, the RAM2 rat develops heart failure, and again, this is attenuated by the use of the GAL3 inhibitor or the ACE inhibitor. In the left lower panel, on the right lower panel, there's again the measures of the invasive hemodynamic measurements, and there's clear indication for impaired relaxation in the RAM2 rat, and that's repaired by using the GAL3 inhibitor. And here again is the pictures of the fibrosis score. In the panel A, the right upper panel, you can see the absence of fibrosis in the control rats, the severe fibrosis in the hearts of the RAM2 rats, and the inhibition of the GAL3 inhibitor, and that's quantified in panel B thereunder. And in an exploratory analysis, as you can appreciate in the Kaplan-Meier survival curve, there is considerable mortality of the RAM2 rats. These are very sick animals with overt heart failure, and it seems that the use of the GAL3 inhibitors is associated with less mortality. So to further study the transcriptional changes that might underpin the mechanisms involved in the antifibrotic effects of galectin-3 disruption and pharmacological inhibition, we conducted gene profiling. We assayed all murine and rat hearts using Illumina gene array chips that contain over 20,000 transcripts. We observed clearly that there was gene expression that's typical for alveolar remodeling, for example, increases in AMP and BMP and all kinds of extracellular matrix genes, but we could not identify clear genes or gene clusters that were activated or deactivated when we disrupted or inhibited galectin-3. And from this we concluded galectin-3 acts not as a transcription factor. So in conclusions, the galectin-3 disruption and inhibition in our hands attenuate adverse cardiac remodeling. It upholds cardiac function and is associated with attenuated cardiac fibrosis. And we believe these preclinical studies validate galectin-3 as a pharmaceutical target and suggest that carbohydrate drugs using the carbohydrate recognition domain of galectin-3 may provide candidates for galectin-3 inhibition. And with this, I'd like to, I think first we have a polling question, so I'd like to give it back to you, Ben.
Thank you very much. Yes, second of 3 polling questions, and we would like to know, let me just put this up here. The recent knowledge of these pathways are likely to spur interest in disease-modifying therapies in heart failure. For those of you in the audience, is your company looking at this? And just click the yes or no circle, click the black submit answer button, and we'll give you a moment to get your answers in. So, Dr. DeVore, how confident are you that the animal model will translate to humans?
Yeah, that's a very important question. I'm pretty confident, though. So, first of all, the sequelae of events in the mouse and the rat model that we used closely resemble the sequelae of events that we see in human heart failure. So it goes from left ventricular hypertrophy to more severe damage, diastolic dysfunction, and ultimately culminating into systolic dysfunction and overt heart failure, dyspnea, fluid overload, and ultimately death. So the models closely resemble the human situation. That's one. And another very important thing to take into consideration is that established therapy, like for example ACE inhibitors or aldosterone blockers, have been tested in these models as well and have been shown to be beneficial, like they are in human heart failure. So also from a treatment perspective, these models are very useful. Well, of course, there's always differences between mouse, rats, and man. I will not deny this, but in general, I think these are fairly well described and established models and very useful for studying pathophysiology and the usefulness of novel treatments.
Excellent. Well, let's see what percentage of our audience is looking into these disease-modifying therapies. Well, we've got a little over 41% are looking into it, which means about 58, 59% aren't at this point. And that's interesting information, and I'm sure you'll be able to use that and see what the market looks like out there as you continue on your research. Okay, so let's continue with the presentation. And Dr. De Boer, why don't you introduce our next speaker?
Yeah, I'd like to give the word back to Peter Munson then, who's President and CEO of BG Medicine. Peter?
Thank you so much. Also a very series of very elegant experiments. Well, sort of step back, I realized that quite a few of the participants were not familiar with galectin-3 prior to joining this conference. I'll talk briefly about what we expected when we started our involvement with galectin-3 now approximately 5 years ago. We expected that patients with high levels of galectin-3 would have this progressive fibrotic form of heart disease, and because of the inherent progressive nature would have a worse prognosis than those who have low levels of galectin-3. We have now measured galectin-3 in approximately 20,000 subjects across several large heart failure trials and consistently found that to be true, meaning high galectin-3 is associated with a poor prognosis when compared with comparable patients with low levels of galectin-3. The second thing that we expected is that galectin-3 patients with high galectin-3, because of the fact that they really have a different disease going on, would respond differently to most drugs. And we have studied that in two large prospective trials and have fully corroborated that in those trials where the benefit of these two different drug classes was completely limited to one group, in this case, the low galectin-3 group. And you see these charts here on the right-hand side. I should add that neither of these drugs are known to be, they're not aldosterone-related medications and don't inhibit the fibrogenesis pathway. The third thing that we expected is that one could inhibit galectin-3 by using a carbohydrate inhibitor that binds to the carbohydrate recognition domain. And although that had been demonstrated in conditions other than heart failure, with the experiment that Dr. De Boer just reviewed, we have now also supported this particular assumption, namely that a carbohydrate inhibitor, and I should add that this is a natural compound, a natural carbohydrate inhibitor, one gets near complete blockage of that pathway with a natural compound, natural carbohydrate inhibitor that binds to the carbohydrate recognition domain. And the fourth thing that we expected is that the galectin-3 elevations, consistent with the comments that Dr. Pitt made early, occur before heart failure is first diagnosed, before signs and symptoms are present, meaning it is the condition that at some point leads to clinical heart failure. And we have also found that to be true. Data on one of the studies was presented at ACC earlier this year in one of the TIMI trials, and last week Professor von Veldhuizen from the University of Groningen, who collaborates with Dr. De Boer, presented on a large epidemiological study that to be true, namely that elevated levels of galectin-3 in the non-heart failure population are associated with an increased risk of cardiovascular death. So we had four important assumptions, and each of these have been validated in particular over the past 12 months. Now, so going back to the slide, we think that there are three important reasons why this galectin-3 story is important for the pharmaceutical industry. One of them is that drugs are unlikely to work equivalently in low and high galectin-3. I'll come back to the regulatory angle of that on the next slide. The second one is that now with the association of galectin-3 with a particular pathway, galectin-3 becomes a target in itself, and obviously having a building on off switch creates an extra opportunity to pursue this as a target because there's a series of natural or synthetic drug candidates identified. And the final one is the point that I pointed to and that Dr. Pitt also pointed to, is that although improving heart failure treatment is a great objective, the even bigger objective that this might enable is finding people before their heart failure, treating them then, treating them at that time, and preventing heart failure from first occurring. And for example, a clinical paradigm would be to take patients at high risk for heart failure development, your post-ACS patient, for example, chronic hypertension patient, patient with diabetes, and periodically monitor for markers like galectin-3. And once elevation occurs, treat that individual with an effective treatment. Well, you mentioned that I've shown in the prior slides these two drug response curves. I didn't point to them, but what they show is that the the benefit of two independent drug classes is exclusively confined to the low galectin-3 patients without any demonstrable benefit in the high galectin-3 patients. And we recently, earlier in May, had a meeting with FDA, great interest from the FDA in this matter, together with one of the leading pharmaceutical companies, to discuss the implications of these findings. Is there to hear the FDA's view on these findings, but also to discuss a possible path forward. I was pleased and to some degree surprised by the very strong statement made by Dr. Stephen Grant that I have tried to paraphrase here. There was something along the lines of heart failure is not a disease. We should no longer indicate drugs for the heterogeneous broad population, but for well-defined subpopulations where we can demonstrate a marked benefit. And I think he then went on to say that it's not an 8% reduction in endpoints in 5,000 subjects they were looking for, but more a 30% reduction in a better defined subgroup. It's, by the way, on Saturday last week at the European Society of Cardiology heart failure meeting, Dr. Ken Dickstein opened his presentation with a slide that said heart failure is not a disease, and something very similar to what is in the quote here. I have recently, with all the attention to hepatitis C, started to compare this galectin-3-mediated fibrosis with sort of the hepatitis C of heart failure. What hepatitis C became for hepatitis for liver disease, a very common, serious, previously unrecognized. When I was in medical school, the term non-A, non-B had just emerged. And in 1989, there was the recognition of the hepatitis C virus, and it enabled a new treatment paradigm, and that resulted in two highly effective curative drugs that have now come to the market that will change the face of hepatitis C as one of the most prevalent and most significant liver diseases in the world, affecting over 258 million individuals. So we think that this galectin-3 finding may put heart failure on a similar trajectory as what the recognition of the hep C virus did for liver disease. On the next slide, one very briefly talk about this pre-heart failure population again. What we have seen biologically in the studies is that the mechanism is there's an injury that could be an acute injury or a chronic injury. I phrase it as the heart gets dinged at some point. There's a link to, for example, Western diet and risks, hypertension, low HDL, diabetes, post-heart attack. These are the kinds of things that chronically or acutely damage the myocardium. It results in an inflammatory reaction, in a secondary fibrosis reaction. The galectins in nature are playing a integrity protection role. You can find galectins in all multicellular organisms where it has been studied. You can find it in worms. You can find it in all mammals where it has been studied. And the consistent role that they play is the protection of integrity. And in this case, you get a fibrotic reaction to protect against acute or chronic injury. But given the longevity of the human race, we are now surviving most of these injuries, and now the inherently good mechanism that might have played a role at the time that we had massive injuries for which we had no medical treatment, so it was a good mechanism in those ages, has now turned against us, and this has become a chronic fibrotic syndrome that causes these secondary negative effects. We have looked at galectin-3 around acute injuries, for example. It is not elevated at the time of an acute injury. It's very different from many of the other markers that we know in heart failure that could rapidly go up at the time of ACS, for example, or an acute decompensation. Galectin-3 is unaffected by those, and the increases typically are observed in studies that we have done that starts at 6 to 12 months after an acute injury. So the model would be that post-heart attack patients would be monitored with periodic galectin-3 samples, and if they are elevated, become elevated at some point, would be a candidate for treatment. And the point that I didn't make before, the work that Dr. De Boer showed is unpublished work. It's expected to be published later this year. We wanted to give the pharmaceutical industry this preview, and many of the references that are underlying the statements that I've made are also studies that are currently on their way to publication. There's a fairly rich literature that we expect to become available later this year. We've also engaged, we're also working in this partnership with the Framingham Heart Study, and are currently measuring galectin-3 in all of the Framingham Heart Study offspring subjects, again, to look to further document this role of galectin-3 in the pre-heart failure population. When you go through the literature on galectin-3 and galectin-3 inhibitors, you will find references to this cardiac, and again, that is fairly recent and evolving. But you will also find use in the liver disease, and there might be an opportunity to treat certain form of liver fibrosis with galectin-3 inhibitor. And there might be an opportunity to treat certain forms of cancer, prevent cancers. Important thing to consider is that galectin-3 acts sort of as glue in the biology and this protection of integrity, if you like. And one of the roles that it plays in cancer, it acts as glue for metastases. And if you block in animal experiments, you block galectin-3, you markedly inhibit metastases formation. What I forgot to mention, consistent with the 50% that Dr. Pitt mentioned, about 30 to 50% of the typical heart failure patients have elevated galectin-3. So it's a fairly common form of heart failure that appears to be mediated by galectin-3. If one starts thinking about how do you interfere with galectin-3, how do you modulate galectin-3, we are currently expanding our screening program to include synthetic carbohydrates purified natural carbohydrates, certain mixtures, to really understand if we can come up with sort of a structure activity definition for these carbohydrates that have this benefit. But obviously, given the fact that it's a protein, antibody strategies are probably going to be highly effective as well. And there are additional other small molecules that could be pursued as specific anti-galectin-3 therapies. Important also, the final point to make is that we know the important relationship between aldosterone blockers and galectin-3. Last Saturday, Dr. Fayez Zanat from France presented some of the preclinical work showing that aldosterone increases galectin-3, aldosterone blockers block that increase in galectin-3, etc. And aldosterone blockers are probably the key drug available right now to block the galectin-3 related damage on the myocardium. I want to summarize it here. Galectin-3 mediated heart failure is a common and serious form of heart failure. We really should stop thinking about heart failure as an indication and as a disease, but really similar to what happened in liver disease, focus on these subforms. In this case, galectin-3 mediated heart failure. The galectin-3 measurement, which will be widely available using these automated assays, can identify those at risk for this disease ahead of signs and symptoms, opportunity to intervene early. Galectin-3 has this built-in carbohydrate switch, excellent opportunity for drug discovery and development. This is not only a situation where we have a good biomarker that can identify the subgroup that is likely to respond, but also have a built-in way to modulate that particular pathway. And that the next point is that even if parties are developing drugs in using other mechanisms in heart failure, it probably is very important to know what form of heart failure one is dealing with. Very similar to what happened in liver disease. The moment these assays became available, the focus was no longer how do drugs work in liver disease, but it became the question, how do they work in a particular form of liver disease. Also want to make the final point that heart failure is galectin-3 mediated heart failure, approximately 50%, 30 to 50% of the overall heart failure population, makes it as significant as most of the other, as most common cancers combined. It's an incredibly important, incredibly large population with a mortality that is comparable to advanced cancers. It's a very common, very serious condition, making it attractive for pharmaceutical development. Well, I'll end the presentation here by talking about what is it that we want to do with the pharmaceutical industry. We are focusing on the diagnostic. We are seeking collaborations with biopharmaceutical companies to support discovery and development of drugs that modulate this pathway. We think that there is a natural win-win between our definition of the patient population and the opportunity to intervene. Various structures, for those who are familiar with the company, we have, over the history of the company, been very successful in structuring partnerships with pharmaceutical companies, with academia, with the public sector, and there are great opportunities to do that here. And we prefer to do that in a non-exclusive way, but have been approached by a number of companies to go down a more limited exclusive pathway as well, and all of that is open. And for those who want to contact us, Rosalie, we can be searched through Google if you like. The email that we have set up is rx@bgmedicine.com. And with that, I want to give it back to Dan, who will lead the question and answer.
Okay, so first we have the third polling question of 3, and what we'd like to know from the audience is, did this program pique your interest in galectin-3 as a target? So we'll just give you a moment to click on the yes or no and then hit the submit answer button. And while we're waiting for that, Dr. Muntendam, do you have anything else that you want to add before we head into the Q&A session?
I think that the key surprise that has become very clear in our minds is that you have this incredible discrepancy between the amount of attention for a particular medical problem and its overall importance. I started saying that heart failure, to begin with, it's a condition that is responsible for more loss of life than all common cancers combined. It is responsible for more hospitalizations than all common cancers combined. And if you go to the Heart Failure Society of America, which is sort of the ASCO for heart failure, if you like, you will find probably fewer people than for any of the cancer subgroups. It's an area that has very little attention. Tremendous. Last time I presented on cancer drug development, there were 512 new agents under development on the list on the statistics that I showed, and now you have heart failure that is numerically more important. It is a higher mortality rate than cancers, and it is virtually not touched by the pharmaceutical industry as an area of new discovery and development. Well, we hope that by pointing to this mechanism, the hep C of heart failure, if you like, that we can engage with the pharmaceutical companies, the biotech companies, the venture communities to really spur the development, discovery and development of new agents.
Okay, thank you. Well, let's see if today's presentation was able to generate any additional interest in galectin-3. And fortunately, 90% of you said that, yes, we were successful in that endeavor. So we appreciate everybody participating in our polls today. Well, throughout the presentation, we've heard and seen a lot of great information. The business and research landscape is constantly changing, and this information will certainly help us stay on top of the key issues. But we've only scratched the surface on this complex topic, so please make sure you send in your questions if you want to go into greater depth on the issues discussed today. It's now time for our Q&A session, which is always a popular segment in our web seminars. We already have many outstanding questions sent in from the audience. Please take a moment now to submit your questions if you have not already. Type your question into the Ask a Question text area in the bottom corner of your screen, then click the Submit button. And please note that due to time constraints, we may not be able to respond to all questions submitted. I would like to reiterate that all registered participants will receive an email within about a week with access to an archive of this live web event. Okay, so let's get to the first question. And this one is for Dr. DeBoer. How does the anti-fibrotic effect of anti-galectin-3 therapy compared to established anti-fibrotic therapy such as ACE inhibitors or aldosterone blockers?
Yeah, thank you, Gwen. Dan, very interesting question. So from in vivo models that we have up until now, the only study that I'm aware of is the one that I shared the data of with you today in the RAN2 rat model, where there was a direct comparison between the galectin-3 inhibiting compounds and ACE inhibitor. But we should remember this is a very peculiar model that is monofactorial, so it's driven by renin and the renin-angiotensin system only. So in this particular model, the ACE inhibitor completely abolishes the phenotype. But even in this model, the galectin-3 inhibiting drug was able to almost completely offset fibrosis. But the very important question, obviously, is how does galectin-3 inhibiting therapy compares to ACE inhibitors or aldosterone blocker, and if given on top of one of these established therapies, does it render further depression of the cardiac fibrogenesis? And these are questions that are addressed in current experimental studies.
Thank you. Okay, our next question is to Dr. Muntendam. What is the percent of galectin-3 mediated heart failure?
Yeah, thank you, Dan. It's as I mentioned earlier, it's about 30 to 50% of the population. It's important to recognize that if one measures it in a particular study, it's important to understand how recruitment for that study could have biased that. For example, some of the studies recruit patients at the time that they present themselves to a hospital with decompensated heart failure. High galectin-3 is a risk for decompensation, so you will find a higher percentage in patients when you enroll them upon discharge than when you take certain all-comers in the outpatient setting. Just to illustrate that further, we have done a study, we've measured galectin-3 in the large HF Action study, large NHLBI-funded study. This was an exercise study conducted by Duke Clinical Research. And in that population, a relatively healthy, broad population of heart failure patients, it's about 30%. And the moment you get to the post-discharge situation, you will find elevations in the 50-60% of the population.
Very good. We're going to stick with you on this one again. Another audience member wants to know if BG Medicine is planning to conduct any further studies related to galectin-3 inhibition.
We are, together with the research facilities at the University of Groningen, with Dr. De Boer and his colleagues, conducting additional experiments with inhibitors, natural and synthetic carbohydrate inhibitors, with two purposes. One of them is to optimize the in vitro and in vivo models that can be used to investigate galectin-3 inhibition. And for example, if we were to work with one of our pharmaceutical partners, or multiple, we would we provide access to that research. It's not funded by any third party. It's funded by us, the company. And there's one clinical study being conducted at one of the top cardiac heart failure research centers in Europe, where we will investigate sort of in a pilot study the use of one of these natural carbohydrate inhibitors to see if we can achieve changes in biomarkers. I should add one thing in the concept of changes of biomarker. Very important concept is that these galectin-3 inhibitors that use the carbohydrate recognition domain do not reduce the level of galectin-3. They turn galectin-3 off. So in the immunohistochemistry done, you'll see that the galectin-3 is still very abundant in the tissue. In other words, it still gets produced, but through the switch, it is turned off and no longer biologically active.
Okay, very good. Did any of our other speakers want to add anything on future research planned? No? Okay. Well, yes, was that one? Okay. We'll go on to the next question. How does galectin-3 relate to some of the collagen turnover markers? You want to start with this one, Dr. Muntendam?
Yeah, and I probably ask Dr. Pitt to comment as well. The slide that Dr. De Boer showed shows that galectin-3 is excreted by macrophages, in turn activates myofibroblasts. Myofibroblasts are responsible for these extracellular matrix changes that lead to fibrosis forming, and the collagen turnover markers are a consequence of that up and down regulation of the collagen turn of the fibrosis. So you see that the galectin-3, in other words, sits at the top of that pathway. The collagen turnover markers sit at the bottom of the pathway, and we think that galectin-3 as the culprit party might have more sensitivity and particular specificity by characterizing that pathway than the use of the collagen turnover marker. So we are investigating how the two relate, but galectin-3 as the culprit is probably where we want to start. But it is very well possible that if a company uses galectin-3 inhibition, that a valuable readout is the reduction of collagen turnover markers.
Excellent. Okay, I'm looking to see what else we have in here. Dr. Pitt, I think I'm going to put you on the spot here, and that's probably going to be all the time we have today. Would you use the galectin-3 dosage in the TopCat patients population, and what would you expect?
Well, I wish we had when we started the study, because if we had, we could have picked out those people at highest risk. Unfortunately, it wasn't available to us when we planned the study. So retrospectively, we could look at it, but we can't do it prospectively. But I think in the future, we'd like to design studies where we prospectively pick people with high galectin and target either an aldosterone blocker or other new antifibrotic strategies to them. So I think we would have been delighted, but it's now unfortunately going to be retrospective.
Very good, thank you. Well, we've hit the top of the hour, so that's all the time we have for our live Q&A. But once again, I encourage you to contact our presenters. They are truly interested in hearing from you. If you still want to submit a question to our speakers, just send an email to webinar2011@bg-medicine.com and put webinar in the subject line. I hope you all enjoyed this event and have taken away some new insights. If any of your colleagues missed this presentation, once again, it will be available as an archive within about a week, and you will be notified via email. Now I'd like to give Peter Muntendam the opportunity for a closing comment. Peter?
Yeah, thank you so much, Dan. I want to thank our speakers, Dr. Pitt and Dr. De Boer, for presenting today. We hope that we have generated interest, and we'd love to speak with each of the companies that have an interest in pursuing this, see if there's a possibility to collaborate to further improve management of heart failure.
Excellent. Well, I'd like to once again thank our friends from VG Medicine and our panelists. Our presenters did an outstanding job. At PharmaVoice, we are always looking for better ways to bring you quality events that address your key issues. You can help us with this by filling out the short survey you will be receiving via email. We also encourage you to look for future web seminars by visiting www.pharmavoice.com. We want to thank you for investing your time in our web event. If you have any questions resulting from today's webcast, please contact us via email at webinar2011@bg-medicine.com and put webinar in the subject line. Thank you for your time and enjoy the rest of your day. The event is now over.